All right, guys, we're just going to hold off just a little bit while we're waiting for people to join us. So we'll just give it a couple of minutes. If you haven't got your data sheet out yet, that might be helpful, as well as a pen and a piece of paper. So if you can run and grab that while we're waiting for people to join the meeting. Because we're going to need our data sheets today. So if you have your data sheet handy and a piece of paper and a pen. I did revamp the notes just a little bit in order for us to try to do this and give you guys 3 hours of work a week, which I don't know how the heck that's going to work. But in order for that to happen, I'm going to have to go through some stuff a little faster and maybe get rid of some of the fluff, even though it's still part of our outcomes. Lucky for you, you're not writing a diploma, so we have a little bit more flexibility. As far as unit exams go, I don't believe I'll be giving any unit exams. We'll work on quizzes, and we'll be doing quizzes, and I might give like a bigger unit end quiz, but they'll all sort of have a very similar weighting. As far as what that weighting will be, we're still kind of in discussions as a science department, but I'll let you guys know as soon as I know. Okay. Probably before the end of the week. All right. So I got a message not being able to hear the audio. If you guys could just put in the chat a yes if you can hear the audio. Okay. So Keenan, there must be something going on your end. I don't know enough about the phone app in order to do that, but maybe look at taking off your audio for 1 second and seeing if that works. All right, so looks like everyone who's going to be here is here. So what we're going to cover today is protein synthesis. So we're going to take a look at that, see how all of that goes. So I'm just going to get a screen ready here for you. Which I probably should have done earlier, but you're going to have to just sort of be patient with us. Your teachers are learning a whole lot of new technology all at once, and it's a little overwhelming, but we are all doing the best that we can. So just so, why do I have a bunch of stuff like that on my drive? That's just weird. There we go. Sorry, just trying to access my stuff. So yeah, as I said, just be a little bit patient with us. Okay, so I'm just going to open up my PowerPoint. So I've made an online version, so this is slightly different than the version that you would have received if you got the PowerPoint already from me. So we're just going to go through this. I'm just going to share this with you on Zoom. 1 second. There we are. All right. Okay, so here is our PowerPoint slideshow, and okay, so molecular biology. So what we're looking at is protein synthesis. So we did go through the first couple of slides of this already. I'm just going to move my annotate bar to the top here. So what we need to understand with this is that proteins are made up of a lot of different amino acids. Okay, so we have 20 different amino acids, and if you're looking at your data sheet, you can see on there that there's 20 different ones, but we'll get to your data sheet in a couple of seconds here. Okay. Chains can be really small or they can be really long. So we can have 8 amino acids or 50,000 amino acids. And what I was saying to you guys in class is what's important about this is how those amino acids come together really affects how the protein folds, and how the protein folds affects its job, because our proteins work by attaching to things. And if they're folded incorrectly, they can't attach to things. And a protein that's really long, that's 50,000 amino acids long, can still have the wrong fold if one amino acid is incorrect. And that can make it so that protein doesn't function. So that's the difference between someone having sickle cell anemia, where their red blood cells can't hang on to oxygen very well, to someone who has normally shaped hemoglobin. Okay. All right. So we'll just keep moving on here. Proteins with 2 amino acids are dipeptides. Okay, and we'll use that term. And then polypeptides, lots and lots. So you can use the term protein or polypeptide. Those terms are pretty much interchangeable. Okay. Proteins are important to us. They're the structural component of our cells. And then we also have functional proteins, enzymes, antibodies, hormones. So all of these have functions or a job to do in our body. All right, so I went through this already with you guys, so I'm just going to go through this one more time. I just want to move this little bar out of the way here a little bit. All right, so what we need to understand with protein synthesis is that there's 2 parts to it. There we go. So the 2 parts to protein synthesis are called transcription and translation. Okay. So over at the top here, just going to move that out of the way, we see this word transcription, and then over at the bottom we see translation. So this is a 2-step process. I'm just going to get a little pencil out, draw myself a picture here, and we'll draw in red. Okay, so we have transcription up here, and we have translation down here. Transcription happens in the nucleus, okay? And translation happens in the cytoplasm that we see there, all right? I'm just gonna add a student here. There we go. All right, and what's happening up here in transcription is basically our DNA only opens up at a gene. So we can see right here, looking at that image, our DNA is going to open up at a gene, and that gene is going to get copied into a different type of nucleic acid called messenger RNA. And once it's copied, then that DNA just closes. It zips right back up. So it opens up, but only at a gene, not the whole DNA, and then it closes right back up and copies that message into mRNA. And then that mRNA moves its way outside of the nucleus and into the cytoplasm. The DNA zips back up and then stays in the nucleus. So our DNA doesn't leave the nucleus. That's where it's kind of safe. So that's its safety zone. So mRNA copies that message and then moves it out into the cytoplasm. That mRNA is going to make its way to a ribosome. So ribosomes are organelles you learned about in Science 10. They're responsible for protein synthesis. And then these other little structures that we see over here, these are called tRNAs or transfer RNAs. One second, just trying to admit. Oh, can I do that? No, I've got to switch to a mouse. There we go. All right. So these little transfer RNAs that we see here, notice if you look down, and this is really small writing, but I can see on the mRNA, I've got 3 U's, and then on the tRNA, I've got 3 A's. So they bind together, okay? And every tRNA carries an amino acid, and it's going to be bringing that amino acid down here to hang to the ribosome, and eventually we're going to get this chain of amino acids, okay? So this whole process of making this chain of amino acids is called translation. All right, so we'll move on to the next slide here. All right, so RNA, we talked about this already. So it's a type of nucleic acid. Okay, so DNA is a type of nucleic acid. RNA is a type of nucleic acid. It's called ribonucleic acid. It's single-stranded instead of double-stranded. The nucleotides have a sugar called ribose instead of deoxyribose. So DNA has a sugar called deoxyribose, RNA has a sugar called ribose. And then the base uracil is used in RNA instead of thymine. Okay. All right. So one thing that I find is a bit of a misconception with students, I'm just going to go to the whiteboard, and I did this with my little practice session with this as well. But let me just get a drawing here and some blue. So when I think of DNA, DNA is double-stranded, but RNA is single-stranded, but it's still made up of nucleotides, right? So I could have a nucleotide of adenine. Adenine's connected to a sugar, which is connected to a phosphate, okay? And I could have a nucleotide with uracil. Okay, I'm just going to see if I can get these people. Oops, cancel. Okay. I could have uracil, and uracil has a sugar and a phosphate. And we could have cytosine, which has a sugar and a phosphate. Okay, so I have 3 different nucleotides, and then these nucleotides aren't joined with a hydrogen bond to make RNA, but the phosphate and sugar in the backbone is joined. Okay, so when I look at this, this is right here a small little piece of RNA. So it's single-stranded, meaning that I don't have nucleotides sitting on the other side that I would with DNA. That's not to say that these guys aren't sticky. They're still sticky. They're still waiting to bond to something. And that's what allows us to create proteins and to create another DNA molecule. Okay, so we're just going to go back to the slideshow here. Okay. All right. Next. Oh, come on. Here we go. So mRNA is a copy of a DNA sequence just for a specific gene. So remember, mRNA only copies a gene from the DNA. It doesn't copy anything else. The mRNA carries the genetic code out of the nucleus and into the cytoplasm. The mRNA or proteins are synthesized at the ribosomes. Okay, so it's going to now go from the nucleus to the ribosomes where proteins can be synthesized. tRNA is a structure that has 3 nucleotides. So when we think about a tRNA molecule, we want to think about something that's sort of, whoops, I'm just going to go back forward for a second. Like this. So when I think about a tRNA, I can think about a structure that kind of looks like this, sort of a shape of a T, and it has 3 nucleotides. So let's say a cytosine, a guanine, oops, well, that looks more like a cytosine, and let's say a uracil, okay? And then that's specifically going to be attached to some kind of amino acid, okay? So that tRNA has 3 nucleotides on it. tRNA is only found in the cytoplasm, where we know that mRNA is found both in the nucleus and in the cytoplasm. tRNA binds to specific amino acids found in the cytoplasm, and we'll talk about how we figure out what amino acids those are. And tRNA carries amino acids to the mRNA that's on the ribosomes, okay? And that's what gets our chain of amino acids to be produced. And then our last one, so first thing with this last one here is you don't have to memorize this. So you can just make a note to yourself. I'll make a note up here. Don't memorize. All right. You don't have to know this one, other than that there is a different structure of RNA, but I'm not going to test you on this at all. But basically what this is saying, is that we have RNA that makes up our ribosomes as well. Okay, but that's not a form that you need to be aware of. Okay, so let's go on to the next slide. And I'm just going to open up the chat here. And like so. All right. So we're just going to fill in the blanks here. So I'll just get you guys to type in what goes next. If DNA is double-stranded, what can we say about RNA? Good. So fundamentally, we need to understand, right? DNA is single-stranded, or sorry, DNA is double-stranded, so RNA has to be single-stranded. Fantastic. Okay, DNA is found in the nucleus, also in mitochondria of animals, plants, chloroplasts. We know all that already, right? RNA specifically is found, and this one I'll help you with. So RNA is found throughout the cell, right? So mRNA is only found inside the nucleus. Sorry, mRNA is found in the nucleus and in the cytoplasm, where tRNA is only found in the cytoplasm. But collectively, our RNA is found everywhere in the cell. Okay. All right. Looking at the chat still. Whoops. Let's close that off here. Again, you're gonna have to bear with me as I'm slowly learning how to do all of this. There we go. Oh, goodness. Doesn't want to do that for me. There we go. Sugar is deoxyribose in DNA, so quickly type in what the sugar is for RNA. Excellent. Okay, good. So ribose is the sugar that we find in RNA. So again, one of the fundamental differences. Okay. All right, thymine is a nitrogen base that's found in DNA. It's not found in RNA. So what is the name of the nitrogen base that's found in RNA? Good, awesome. Okay, DNA, we have one type. We just talked about how there's different types in RNA. How many different types did we just talk about in RNA? Good, awesome. So there's 3 types of RNA. And I'm just going to slide this up here. Okay, DNA. Now we have to remember DNA, that's it. That's all of the nuclear material. I just got a question, what's uracil? Uracil is a type of nucleic or nitrogen base that is only found in RNA. It's not found in DNA. And it is on your data sheet as well. Okay, so it's just a different nitrogen base, and it acts just like thymine would act. So thymine would join to an adenine. In RNA, uracil will join to an adenine. Okay. All right, so DNA is really, really long. Because RNA is only either like the 3 base nitrogen bases of tRNA or a gene, what could we say about the length of RNA? Good, so RNA has to be shorter. Fantastic. Okay, so you guys are getting this. Okay, let's move on to the next slide here. Okay, so there's 2 steps in protein synthesis, like we talked about, transcription and translation. So transcription, like I said already, is what happens in the nucleus, okay? So transcription is just that DNA opens up just at a gene, and it copies all of that information into an mRNA. So the DNA message of a gene gets copied into mRNA. Now, the thing we have to remember is that DNA has 2 strands, where mRNA is just 1, which means mRNA is only copying 1 strand of the DNA. Okay. All right. Transcription happens in the nucleus. Okay. We must know that absolutely for sure, right? This is like a star. Whoops. Get good at this here. This is a thing we want to star, right? Happens in the nucleus. We must know that. Ooh. Okay, I must have paused that for a second. Can you still see my screen? Open. Okay, go back to my mouse here. Okay, awesome. Thank you, guys. All right. So what happens with transcription? DNA unzips at the gene. DNA unzips at the gene of the DNA that codes for a specific protein. And then we have an enzyme called RNA polymerase. So we learned about DNA polymerase, which helps to build that second strand of DNA. RNA polymerase builds that strand of mRNA. Okay, so building a polymer, polymerase, it's an enzyme. All right, so when we look at this I really like this image because it gives us a really good sense of what's happening. A couple of things that we want to take note of. When we are trying to figure out what the mRNA sequence is for the DNA, we will always use the given, sorry, I didn't bring my special pen here, use the given DNA sequence. To make mRNA. Okay. So what I mean by that is oftentimes in a question, you'll just be given one strand of DNA. That's the strand you make into mRNA. Okay, so that's the protocol that we'll follow. Even though you're not writing a diploma, you're still writing questions for me. So always follow that protocol. All right. Okay, so now let's take a look at what this image is telling us here. So here's our gene. Whoops. Go forward here. Here's our gene. So I'm just going to circle that in blue there. Okay. Now what we have going on is our DNA is opened up at that gene. And as it's opening up, here we see the transcribing strand of DNA. And we see these little nucleotides, so free-floating nucleotides, just like what are used to make mRNA. These free-floating nucleotides are going to attach to that DNA. And they're going to continue to attach until we've got that whole gene copied. And when we've copied that whole gene, then that mRNA gets kicked off. All right, and then I have a little thing here to add. There we go. Thing to remember is that the whole entire molecule of DNA does not unzip when we're looking at transcription. It's just where the gene is that unzips. Okay, so it just unzips at the gene, and here we see this nice little sequence being formed. Once that whole transcript of mRNA is done, then this mRNA gets kicked off. So let's just draw that. This mRNA gets kicked off, and that DNA is going to come right back together. Okay, so the DNA is going to stay inside the nucleus, and what is going on here? There we go. And I want you guys to just quickly tell me, where's that mRNA going? Okay, it gets kicked off the DNA. Where does that mRNA go? Put that in your chat. Awesome. So that mRNA is going to make its way to the cytoplasm. Okay. And what organelle is that mRNA going to attach to? Awesome. Okay, so that organelle that the mRNA is going to attach to is the ribosome. So again, back in Science 10, you learned ribosome's a site of protein synthesis. So that's what we're talking about. Okay, perfect. All right, so now let's move ahead here. Let's make this go. All right, so once the DNA or the gene is copied, mRNA is released from the DNA. DNA strand zips back together. mRNA moves out of the nucleus to the ribosome, which is in the cytoplasm. Okay, which you guys know. Okay. This doesn't want to. All right, we're not going to watch the video. I have this linked so you can watch the video and just get a sense for what transcription looks like. Okay, I want to save our time with just me talking, so I would recommend you going back to watch that. All right, so what we're looking at here And this is where in the notes that I have, you can write what these sequences are, and we're going to talk about it. So let's just look at question number 1. So question number 1, I have a sequence of DNA that is to be transcribed into mRNA. And then I have a sequence of DNA that is corresponding to that transcribing sequence. Okay. So what I would like you to do is, first of all, type in what the corresponding DNA strand should look like. Okay. I will just open up my chat here. Okay. Type in what you think that corresponding DNA strand should look like. Good. All right, so we're looking at a GCC TAT. Nice. Okay, perfect. So it's really good you guys understand how to do that. Now, because I gave you this strand, okay, so there's our right answer. Now we want to make that into mRNA. So now I'd like you to type in, what would the mRNA strand look like? So if this is our strand, the one that I gave you, the one that's in black, if that's the strand that's going to be now transcribed into mRNA, what should that strand of mRNA look like? Okay, good. So I'll just put that up here. Okay. So basically, when we're doing this, I got a question of how to do this. So C's always have to combine with G's, and A's always have to combine with T's, except for the fact that mRNA doesn't have T's. mRNA has uracils instead of thymines. So wherever we would normally put a thymine, so if I look here, right, this, whoops, let's go back a sec. So my spotlight there, that would normally be a thymine if it was a DNA, but because it's an RNA, there is no thymine, so that's where the uracil is going to go. Okay, so the big difference that we notice in this between our corresponding DNA strand and our mRNA strand is that there's U's in the mRNA where there would be T's in the DNA, and otherwise they look pretty much the same. Okay, now I'm going to skip past the second example here. If I can. There we go. All right, so now we're going to move into translation. So translation is the formation of a protein, which is a chain of amino acids. Okay, so this is where this gets just a little bit more tricky. Okay, so we're going to use the code on the mRNA to figure this out. So translation, as we've already talked about, this happens in the cytoplasm. mRNA inserts into the ribosome like a ribbon. So basically we have our ribosome, our mRNA, and it's moving along, okay? So our ribosome is going to move along that mRNA strand, and then we're going to end up having the formation of a protein from that, okay? All right, so let's look at how this works here. So the first thing we need to do, and what I would like you guys to do, is just take out your, if you have your data sheet available, if you can take that out. And I am going to just get a new share going for us. Just give me 1 second here. So I didn't bring this up like I should have already. But we will find it. As you can watch me sift through all of my stuff here. And sorry, wasn't quite as prepared as I wanted to be. Okay, just going to bring out this data sheet here. All right, so if you have your data sheet handy, this is what I want you to do with your data sheet. I want you to circle about 1,000,000 times here, messenger RNA. And there's a point to that. There's actually 2 points to that. So those of you who have taken or are taking Science 30 do not have this data sheet. Your data sheet says DNA codons, okay? In Science 30, a little bit of magic happens and mRNA doesn't exist, okay? Our data sheet says mRNA, but it only says mRNA. It doesn't say DNA, it doesn't say tRNA, it says mRNA. So we have to be able to use this data sheet with mRNA, but mRNA only, okay? All right, so have this out in front of you, and I'm going to just change what I'm sharing here and go back to my slideshow. All right. There we are. Okay, so codons are 3 nitrogen bases of mRNA that code for a specific amino acid. So what I'd like you to do is look at your data sheet and then type in the share and tell me, what does UUU code for? Okay. What I was circling, Jordan, is just the title of your data sheet that says mRNA. So on the data sheet, there's a table that says mRNA. Circle that part over and over again that says mRNA, because that's the part that we need to have. Okay, awesome, you guys. So you got phenylalanine from that. Okay, let's just do one more and then we'll go to the next slide. So ACU, so I'd like you guys to type in, what does ACU code for? Okay, perfect. Okay, so ACU codes for threonine. So that's how we use our data sheets. So we're just looking stuff up. But remember, only good for messenger RNA. All right, that's it, just messenger RNA. Okay. A couple other things I need to explain to you. So value of value of redundancy. So what does this mean? I'm going to just jot a couple things down on the whiteboard just to help us to understand what I mean when I talk about value of redundancy. We'll just clear this. Okay, so value of redundancy, if I have something like, let's say I should have my data sheet up in front of me here as well. Okay, so if I have something that says A, so I have a codon that says AAA, all right? And I know then that that's going to so that's, sorry, this is my DNA. That's going to transcribe into mRNA, and that mRNA is going to be UUU. Okay? And then that, when it's translated, it actually codes for the amino acid phenylalanine. Okay? But let's say a mutation happened in the DNA. And let's say the mutation, so we now have a mutated DNA. Okay? And in our mutated DNA, we have AAG. Okay, so a mistake was made. So this G is wrong. So that's a mutation. DNA is wrong. So that means we have AUUC. Now, what I want you to do is look on your data sheet and tell me which amino acid is coded for with UUC. Matika, the codes confuse me. So the data sheet, it is on the data sheet, so you have to look at your data sheet. I do have it on my website, so if you don't have one with you or you don't have it at all, you can copy it later from my website. Okay. All right. So if you can take a look and tell me on your data sheet, what does UUC stand for? What amino acid? Hey, phenylalanine again. Yeah, okay. So when you look at your data sheet, what you notice is that there is more than one code for some of the amino acids. So if a mutation happens, there's the possibility that the same amino acid would be incorporated into that protein, which means the protein won't have the wrong fold and the protein will work. So that's where we get this benefit of having this value or benefit of redundancy. Okay, so let's take a look here. So yeah, it's one way for mutations not to happen as often as this. So this is referred to as what's called a silent mutation, where we can have a mutation, but it doesn't have any change to our phenotype. Okay. All right, so I'm going to go back to the slideshow here. And we'll just look at this value of redundancy. So what I did was just a little bit of math, right? So my 4 to the 3, what that really means is, let's move that up there, is if I have 3 possible codons, right? And I've got 4 possibilities for each codon. It could be an adenine, a thymine, or sorry, an adenine, a uracil, a guanine, or a thymine, guanine or a cytosine, sorry. Then there's 64 different possible combinations of codons. And if you look at your data sheet, there are 64 different combinations, but there's more than 1 different codon for amino acids. And that's why we have so many different combinations, okay? So when we think about that value for redundancy then, right, why is it important to us? If a mistake is made in the codon, it leads to the incorporation of the same amino acid. Now, one thing I want to just take note of here, one common misconception is that this process of translation is building or creating amino acids. That is not correct. All this process is doing is taking these free-floating amino acids that are attached to tRNAs and assembling them, okay? All right. So we'll just go on to the next slide here. Okay. So initiator codons are start codons. And if you look at the bottom of your data sheet, you can see where your initiator codons are. So your start codon, and then your terminator or stop codon. And I'm just going to go back to my data sheet just so I can show you where those are. I can find my data sheet here. Okay, sorry guys, I have way too many windows open. There we go. Okay, so if we scroll this data sheet, what we see here is our initiator codon, so that's AUG, and then our terminator codons are here, UAA, UAG, and UGA. They're all on the table, so you don't have to memorize those. Those are sitting right there on that table, okay? All right, so AUG is our start codon and then our stop codons. And again, those are just coming right off of your table, okay? All right, so tRNA is a special molecule that is specific to an amino acid and it transports it to the ribosome, okay? So these are carriers of amino acids. They have 3 nitrogen bases that are called anticodons. So anticodons are part of our tRNA, where codons are on our mRNA, okay? Anticodons correspond with codons, so they can attach to one another. Once bonded to the mRNA, the tRNA drops off the amino acid and then leaves the ribosome. So what I want to do is I just want to take a look here. And again, if you have your data sheets, this is helpful. What I would like you guys to do is indicate what the mRNA, whoops, I'm just going to go back here, forward here, I mean. Indicate what the mRNA would be. So here's my tRNA. So remember, these guys are called the anticodons. I want you to figure out what the mRNA codon would be. And then I want you to figure out what amino acid is being carried by each of these tRNA molecules. So let's start off with the first one, okay, since we're sort of stuck to using chat here. Ethan, for the terminator codons, you just have to, you can get them right off your data sheet. So they're right in the bottom part of your data sheet, okay? So if I take a look at this very first, so here's my little spotlight. I'm just gonna highlight this very, oh, it's not working. There we go. Oh, can't do it, sorry. The very first tRNA we see, first thing I want you to do is just chat in what is the mRNA sequence, just for that very first tRNA. Okay, perfect. So UCU. Okay, so let's just put that in there. So we have UCU. All right. Next thing I want you to do now is figure out what amino acid is being carried by this tRNA. So using your data sheet, what amino acid is being carried by the tRNA? All right, so we have serine there. Get my mouse here. Serine. All right, let's do the next one, okay? So our anticodon is CGC. So the first thing I want you to do is what would be the mRNA codon for that? Okay. All right, good job, you guys. So we've got GCG. Now, what amino acid is being carried by that tRNA? Okay, I'm impressed, you guys. Nicely done. Okay, so let's just put this next one in here. Okay, alanine. So the trick with this is when I'm looking at trying to figure out what the amino acid is, I always have to use the mRNA. I can't use the tRNA anticodon. I have to use the mRNA codon in order to figure that out. Okay, why don't we just quickly do the last one here? Okay, so we have, just gonna move this over. So what would be our mRNA for our last? Okay, perfect. And now that we know our mRNA is AAA, what would be our amino acid? All right, you guys are awesome. Okay, perfect. Because that's probably the biggest challenge is understanding that we have to use the mRNA in order to figure out what the amino acid is that's carried by the tRNA. Okay. So if we look over here, there we are. Nope. Same thing. All right, so the ribosome continues to move along the mRNA. As the ribosome, I've just got a question here, difference between start and stop codons. Start codons are what are going to initiate protein synthesis. Stop codons, basically what a stop codon does is it kicks that protein off of the ribosome. So that amino acid sequence that's been building gets kicked off of the ribosome, okay, with a stop codon. So the ribosome moves along the mRNA, so it continues to move along the mRNA. tRNAs continue to drop off their amino acid until we have this chain of amino acids that's being built, okay? The chain of amino acids will grow until we get to this terminator signal or a stop codon. So a terminator signal is the same thing as a stop codon. And then that protein, that chain of amino acids gets kicked off into the cytoplasm. And that mRNA gets released back into the cytoplasm as well and just chopped up into nucleotides to be reused. Okay. All right. So when we're looking at this image here, so this just gives us a little bit of a sense for how this works. Okay. So let me just put up my drawing here. Let's do that green. So here I see my ribosome. So there's the ribosome there. And what happens is these tRNAs, so notice the tRNA, we have this anticodon sequence, and we have this amino acid attached to it. So that tRNA is attaching to our start codon. Now, our start codon actually codes for an amino acid. Our terminator or stop codons do not, okay? So again, we're just gonna play what's that amino acid. So what's this first amino acid here that's being attached to the ribosome? Okay, so I'm looking for this one right here. Ideas? Yeah, good. So we're looking at the mRNA sequence, right, which is the one on the bottom, and we're using that to figure out what amino acid it is, and that amino acid is methionine, okay? All right, and what's the next amino acid? Okay, good. So UUC, awesome. So we're seeing phenylalanine. And then we have this other amino acid, right? So this one hasn't attached yet. So the thing to understand about this is that our tRNAs, they can't hang on or attach to the mRNA until this ribosome has moved along. So this ribosome is going to continue to move. And as soon as this codon is sitting on the amino acid, or sorry, sitting on the ribosome, then this tRNA can attach, but it can't attach yet, okay? And what amino acid is this tRNA hanging on to? Okay, awesome. Oh, you guys are fantastic. Okay, perfect. All right. So what I have here is 3 different videos. Again, I'm not going to watch them with you, but I would really strongly recommend watching them because it really helps with this process of understanding, because you can see the moving pictures, and the moving pictures make this a lot easier to understand. Okay, I think, oops. All right, so when we take a look at a summary here of protein synthesis, what we're looking at is transcription. Oh, and transcription, if you could write in the chat, where does transcription happen in the cell again? Excellent. Okay, so transcription in the nucleus. And what part of the cell does translation occur in? Okay, good job. So translation is in the cytoplasm. Okay. So all this is showing is a little section of a gene that's being copied, and it kind of just gives us that process, right? So going from DNA being copied into mRNA, that's transcription, and then that mRNA being used to create a chain of amino acids or a protein, that's translation. All right. So one more quick thing we're going to do here before I let you guys go for the day. What we're looking at with this image here is we have another DNA strand. So this is the DNA strand that's being transcribed. Whoops. Okay, that one I'm not too worried about because you guys seem to know that really well. But this is the DNA strand that's being transcribed. And that's being transcribed into mRNA. So you guys already showed me that you know how to do that really well. So I'm just going to show you that. So here's our mRNA strand. And again, what we noticed from what we did earlier is that our other DNA strand, and our mRNA strand are almost identical, except our mRNA has uracils where our other DNA has thymines. Okay. All right. What we're going to do here is we're just going to try to figure out what amino acids are being carried. So the way I want you to think about this is, imagine that our tRNA is kind of upside down. We have one tRNA there, and then we have another tRNA here, and we have another tRNA here, and another one here. And each of these tRNAs, so what I'm making these little boxes for is for the anticodon, and they're all attached to an amino acid. Okay? So I just want you to take like 30 seconds and write down what those 5 amino acids would be. Okay? Let's do just write them down on a separate sheet of paper, what those 5 amino acids would be. So I'll just give you 30 seconds here. All right. So the first amino acid I'd like you to tell me, what is the first amino acid? I'm just going to open this up online. Okay, awesome. All right, so your 5 amino acids should look like this. So we should have serine, histidine, serine, aspartate, and serine. Are we okay with that? Any questions about that? All right, I do have question number 2. I'm not going to do that in class right now. We are going to stop there as far as me presenting. So I have a couple of things for you, and I'm going to post those on Moodle. So the first thing I want you to do is there's a protein synthesis worksheet, and I'd like you to complete that worksheet. The tRNA nitrogen bases. So remember the tRNA, so I'm just answering a question here, guys. So understanding the tRNA. So remember the tRNAs, just get a different color here. Remember the tRNAs are attaching to the mRNAs, so they have to correspond. So remember that tRNA has to attach here. So those have to be able to bond, okay? So for the protein worksheet, I would like you guys to do that and submit it. So I will give you until Wednesday to submit it. So tomorrow is going to be a work day for you guys. On your work day, I'd like you to watch the YouTube videos that we didn't watch. I would like you to do the protein worksheet. And then the DNA quiz, I'm going to open up on Wednesday. So you'll have an opportunity to study for that. And then you'll have 2 days to write the quiz. And we're going to do the DNA quiz just like we did the Moodle quiz. So you have to answer 1 question at a time because you can't go back, okay? And answer the questions to the best of your ability. Yes, Ethan, I am going to post this. Oh, I will post a list of what we need to do. Absolutely, Avery. Yeah, I'll do that after we're done this lesson here. Lecture for tomorrow, no. We're going to do just a lecture on Wednesday and then another lecture on Friday. So Wednesday's lecture will be finishing this booklet and then probably starting our next unit, which is populations, because I'd like you to get started on that over your spring break. And yes, as far as posting the protein synthesis worksheet, you can post it. Now, this is a new worksheet, so if it gave you the old worksheet, you'll have to check that one. The new worksheet's been posted already. How you want to submit it. If you want to do it and submit it as a digital copy and it's all done digitally, that works for me. If you want to print it out and do it and then take pictures of it, that works for me. And it looks like you can actually take a picture of it if you have an iPhone with the notes section. You can take a picture of that and turn it right into a PDF that way as well and send it to me that way. Honestly, whatever is easiest for you guys to submit it to me works for me. As always, just ask me any questions you need to. I check my Gmail like a million times a day these days, so I'm happy to answer any of your questions. I also will have a classroom open just for any questions. So I'm hoping to hold office hours from 9 to 12 for the remainder of the week. Antisense strand. Ashley, is this the worksheet that has a whole bunch of different sequences, the front and back? Yeah, that one I pulled off, so don't worry about doing that one. Just do the other one that I posted. Okay. Yeah, so that's it, everybody. Unless there's any other questions, you also are free to, unmute your mics and ask me questions verbally as well. Okay, I know this sucks, but you guys are absolutely fantastic, and I think this is going to go as good as we can make it go. So I will post everything that I'm asking you guys to do on Google Classroom in the next few minutes. All right. So have yourselves a wonderful day, and if I don't see you in my classroom, or in my question room, then I will see you guys on Wednesday. All right, take care. You're very welcome, you guys.